biology as they helped in laying the foundation of genetics. Questions like, why do kids look similar to their parents? And why are there a few differences in two kids that belong to the same parents? Started gaining answers. We now have understood the monohybrid cross and the monohybrid ratio as well. This is how it's done. Take a moment to review it. How did we get the F1 generation? We got it by considering all the possible combinations here. But is there a method to find it systematically? There is. So before moving ahead with the dihybrid cross and ratio, let's have a look at an extremely important and useful method for writing a cross. The method is named as Punnett square. You may wonder as to why do we need another method for a theoretical cross when we already know how a cross is carried out? Well, that's because this method is quite useful in many ways. The technique was developed by the British geneticist Reginald Punnett. Hence, it's most commonly known as Punnett square. Let's understand the technique step by step first. The first thing we do is make a table like this. We write the symbols of male and female here. These can be written vice versa as well, but for now, we write it like this. This tells us that the maternal alleles will be written here, while the paternal alleles will be written here. So let's begin the crossing now. This allele can come together with this one to give us this set of genes in the offspring. These two can give us this set. This one will combine with this to give us this set. And what about the last one? Yes, you guessed it right. These two alleles will give us this set in the offspring. It's a simple concept of respective row and column pairing for each slot. For this slot, it's the first row and the first column. For this slot, it's the first row and the second column pairing, and so on. Please understand this well, because it's going to help us a lot in the future. Now, if we compare the simple cross that we usually carry out, this is what we get. Notice that the results are exactly the same. All the plants in the F1 generation are tall and have the same genotype. They are all heterozygous. This confirms that our cross is correct. A glance at the presentation of this cross hints at one more name for it. Yes, this technique is also referred to as the checkerboard method. Now, let's do this cross one more time to know whether we've understood it correctly. Let's cross any two from the F1 generation now. Again, we first draw these lines to form a table and write the male and the female symbols here. Now it's the turn of the genes. So we write the respective genes here and here. Shall we begin crossing now? Why don't you try filling in these 4 slots? Here we go. These are the combinations that we obtain on crossing these plants. Take a moment to go through this table. Can you find the phenotypic and the genotypic ratios here? Phenotype, if you remember, refers to the observable physical properties. We can clearly see that there are 3 tall plants and 1 dwarf plant. So the phenotypic ratio is 3:1. What about the genotype? There is 1 homozygous tall case, 2 heterozygous tall cases, and 1 homozygous recessive case. So the genotypic ratio is 1:2:1. Aren't these the monohybrid ratios? Yes, they are. We get exactly the same results that we obtained on crossing the F1 generation previously. So, this is how monohybrid cross can be carried out using the Punnett square method. Can you guess why this method is used when we can cross the alleles directly? Yes, of course we can find it directly. But that's because a monohybrid cross involves the crossing of a single character at a time. Hence, it's easy. But what about the crosses that involve more than 1 character? An increase in the number of genes can make the crossing a bit complicated. Say we have 2 or 3 genes. Then do you think a simple cross like this will be convenient to carry it out? Of course not. It will be a huge mess. So in order to simplify this, we make use of a Punnett square. This method is simple and extremely useful when more than one alleles are involved. Talking about crosses involving more than one character, what are the crosses involving 2 characters called? The cross involving 2 characters at a time is called dihybrid cross, and the ratio obtained will be a dihybrid ratio. Let's understand this interesting concept in the next part. Here is a representative monohybrid cross on our screen, which involves only one character at a time. Do you think crossing a single character at a time was enough for Mendel to arrive at concrete conclusions? Of course not. After all, the 7 pairs of contrasting characters were crossed individually. Mendel went a step ahead and crossed plants considering 2 characters at a time. He tried the dihybrid cross. Let's take a simple example to understand how the dihybrid cross works. Which 2 characters should we take now? We've already seen a monohybrid cross for characters like the plant height and the flower color individually. So let's consider these characters together to understand the dihybrid cross. For our convenience, we take one plant which has both dominant characters, while the other with both having recessive characters. It means we take both the plants which are homozygous. Any guesses about what the genotypes will be? TTPP in uppercase will be this plant which is tall with purple colored flowers. Similarly, TTPP in lowercase will represent a plant that is dwarf and has white colored flowers. This is how we separate the gametes. Any idea why didn't we write it as TT and PP? The alleles T and T represent the same trait, so they will have to be distributed in the respective gametes. They will not get together in the same cell. Similarly, P and P will get into the separate cells as they are the alleles for the same trait, flower color. It's the same case in the other plant as well. This is how the gametes will be formed. Now we're ready for the crossing. This is how the F1 generation would look. Take a moment to understand this. Now, to get a conclusive result, we need to cross the offsprings from the F1 generation and get the F2 generation. Let's pick any from the F1 generation. Can you tell me how the gametes will be formed here? Gametes can be formed in these 4 different combinations for each case. Did you notice what happened? Will it be easy to do the crossing here? Definitely not. That is the reason why we opt for Punnett square for a dihybrid cross. Let's get started with the cross for obtaining the F2 generation. We write the male and the female symbols first. Followed by this, we write the respective gametes here and here. And how do we cross these to fill in the 16 slots? We cross each row and column respectively. Here, this TP is crossed with this TP, so we obtain this slot. Similarly, we cross this TP with TP to get this offspring. Can you try filling in the 3rd and the 4th slots in the 1st column? We cross this TP with this TP to obtain this offspring. And lastly, we cross this TP with this TP to obtain this one. All 4, as we can see in the table, are tall with purple flowered plants. That's because there is at least one capital T and one capital P in each case. If you wish to, you could pause the video to fill in the other 12 slots. On similar lines, these are the results that we obtain in the other slots. Take a moment to go through all of these. Let's count the different combinations now. These are the 4 different possible combinations. Tall with purple flowered plants, tall with white flowered plants, dwarf with purple flowered plants, and dwarf with white flowered plants. Now, can you count the number of plants that are tall and with purple flowers? We have 9 plants which are tall and with purple colored flowers. Can you count the number of plants for the other 3 combinations? 3 plants show the tall trait along with white flowers, and 3 show the dwarf trait along with purple flowers. 6 plants in all have mixed characters of the parents from the P1 generation. It means at least one recessive character is seen in these 6 plants. Now, the last one among the 16 shows both the recessive traits, a plant which is dwarf and has white flowers. So if taken together, then we obtain a ratio 9:3 to 3:1. This ratio is referred to as the dihybrid ratio. Now, Nora has a simple question for you. Whenever any 2 characters are randomly taken for a cross, do we obtain the same ratio? Say we take the seed color and the seed shape. Will we obtain the same ratio? Well, if the cross is carried out correctly, then we are bound to get 9:3 to 3:1 as the dihybrid ratio. So if there are 16 plants obtained in the F2 generation, then 9 of them on an average will express both the dominant characters, like the tall height and the purple colored flowers in this case. 6 plants on an average will express a blend of both the characters, dominant and recessive. Just like these 3 expressing tall height and white flowers, and these 3 showing dwarf height and purple flowers. Lastly, only 1 out of the 16 shows both recessive characters. Do you think we always get exactly 16 plants in the F2 generation? No, not at all. There are numerous seeds obtained, which are then sown, and plants are grown to observe these results. Did Mendel get only 16 plants in the F2 generation? This is also not the case. Mendel had obtained many seeds, but when he grew plants from them, he obtained the plants in certain numbers. When these numbers were calculated, the ratio turned out to be 9:3 to 3:1. So basically, 9:3 to 3:1 is the ratio obtained at the end of the calculations. The total number of seeds, and thus the plants obtained, could be much higher. Do watch our videos based on ratios if you want to understand them better.